Welcome students to a lecture on the topic DNA-drug interaction and combinatorial synthesis. In this lecture, we will deliberate upon the following topics. First, DNA-drug interaction and the various classes of such drugs. Second, basic mechanism of interaction of some selected drugs. Third, we will be discussing antisense therapy and its applications. Fourth, combinatorial synthesis and its types. In the end, we will be discussing the limitations of combinatorial synthesis. DNA as a carrier of genetic information is a major target for drug interaction because of the ability to interfere with transcription, that's gene expression and protein synthesis, and DNA replication, a major step in cell growth and division. The latter is central for tumorigenesis and pathogenesis. There are three principally different ways of DNA drug binding. First, through control of transcription factors and polymerases. Here the drugs interact with the proteins that bind to DNA. Second, through RNA binding to DNA double helices to form nucleic acid triple helical structures or RNA hybridization. That's sequence-specific binding to expose DNA single-strand regions, forming DNA-RNA hybrids that may interfere with transcriptional activity. Third, small aromatic ligand molecules that bind to DNA double helical structures by first intercalating between stacked base pairs, thereby distorting the DNA backbone conformation and interfering with DNA-protein interaction. Or the minor groove binders. The latter causes little distortion of the DNA backbone. Both work through non-covalent interaction. The small ligand drug approach offers a simple solution. The synthesis and screening of synthetic compounds that do not exist in nature work much like pharmacological ligands for cell surface receptors in excitable tissues and appear to be more readily delivered to cellular targets than large RNA or protein ligands. The lack of sequence specificity for intercalating molecules, however, does not allow to target specific genes, but rather certain cellular states or physiological and pathological conditions like rapid cell growth and division that can be selectively suppressed as compared to non-growing or slowly growing healthy tissue. Representative DNA binding drugs. There are various DNA binding drugs, amongst which some have been discussed as DNA intercalators. A typical example of this class is amascrine. Amascrine or amascrine, synonymous with M-AMSA, that is acredinyl anisidine, is an antineoplastic agent. It has been used in acute lymphoblastic leukemia. Its planar fused ring system can intercalate into the DNA of tumor cells, thereby altering the major and minor groove proportions. These alterations to DNA structure inhibit both DNA replication and transcription by reducing association between the affected DNA and DNA polymerase, RNA polymerase, and transcription factors. Amascrine also expresses topoisomerase inhibitor activity, specifically inhibiting topoisomerase II, compares with the better known agent etoposide. In contrast, the structurally similar O-AMSA differing in the position of the methoxy substituent group on the aniline ring have little ability to poison topoisomerase II despite of its intercalative behavior, suggesting that intercalation of the molecule in itself is insufficient to trap topoisomerase II as a covalent complex on DNA. Another DNA binding drug is natropsin, which is a class of groove binders. Natropsin is a polyamide with antibiotic and antiviral activity. Natropsin was discovered by Finlay et al. And first isolated from the Antibacterium streptomyces natropsis. It belongs to the class of pyrolamidine antibiotics, synonymous for the compound or congocidine and cynanomycin. Natropsin binds to the minor groove of adenine-thymine rich sequences of double-stranded DNA. In contrast, natropsin does not bind single-stranded DNA or double-stranded RNA. Crystallographic structures of DNA-bound natropsin have been obtained, and these studies elucidate details of how the drug binds in the minor groove. In the bound structure, the drug makes hydrogen bonding interactions with 4 subsequent base pairs of the DNA duplex, locally displacing the water molecules of the spine of hydration. It has been shown that natropsin is active both against gram-positive and gram-negative bacteria. Another class of DNA binding drugs is DNA alkylators, such as meclorethamine and carmustine. Meclorethamine is an anticancer, antineoplastic or cytotoxic chemotherapy drug. Meclorethamine is classified as an alkylating agent. It works by binding to DNA, cross-linking 2 strands and preventing cell duplication. It binds to the N7 nitrogen on the DNA base guanine. Carmustine, also known as bischloroethyl nitrosourea, BCNU or BICNU, is a medication used mainly for chemotherapy and sometimes for immunosuppression before organ transplantation. It is nitrogen mustard beta-chloronitrosourea compound used as an alkylating agent. As a dialkylating agent, BCNU is able to form interstrand crosslinks in DNA, which prevents DNA replication and DNA transcription. It's used in the treatment of several types of brain cancer, including glioma, glioblastoma multiforme, medulloblastoma and astrocytoma, multiple myeloma and lymphoma. BCNU is sometimes used in conjugation with alkyl guanine transferase, that is AGT inhibitors such as O6-benzyl guanine. The AGT inhibitors increase the efficacy of BCNU by inhibiting the direct reversal pathway of DNA repair, which will prevent formation of the interstrand crosslinks between the N1 of guanine and the N3 of cytosine. It's also used as a part of a chemotherapeutic protocol in preparation for hematological stem cell transplantation, a type of bone marrow transplant, in order to reduce the white blood cell count in the recipient patient. Drugs that form covalent bonds with DNA targets. Drugs that interfere with DNA function by chemically modifying specific nucleotides are mitomycin C, cisplatin, and anthromycin. Mitomycin C is a well-characterized antitumor antibiotic which forms a covalent interaction with DNA after reductive activation. The activated antibiotic forms a cross-linking structure between guanine bases on adjacent strands of DNA, thereby inhibiting single-strand formation that is essential for messenger RNA transcription and DNA replication. Anthromycin is an antitumor antibiotic which binds covalently to nitrogen 2 of guanine located in the minor groove of DNA. Anthromycin has a preference of purine-guanine-purine sequences with bonding to the middle guanine. Remember, purines are adenine and guanine. Cisplatin. Cisplatin is a transition metal complex cis-diamine dichloroplatinum and clinically used as anticancer drug. The effect of the drug is due to the ability to platinate the nitrogen 7 of guanine on the major groove site of DNA double helix. This chemical modification of platinum atoms cross-links 2 adjacent guanines on the same DNA strand, interfering with the mobility of DNA polymerase. Now there is an important topic, antisense therapy. Antisense therapy is a form of treatment for genetic disorders or infections. When the genetic sequence of a particular gene is known to be causative of a particular disease, it is possible to synthesize a strand of nucleic acid such as DNA, RNA or a chemical analog that will bind to the messenger RNA, that's mRNA, produced by that gene and inactivate it, effectively turning the gene off. This is because messenger RNA has to be single-stranded for it to be translated. Alternatively, the strand might be targeted to bind a splicing site on pre-messenger RNA and modify the exon content of a messenger RNA. This synthesized nucleic acid is termed as antisense oligonucleotide ASO because its base sequence is complementary to the gene's messenger RNA, that's mRNA, which is called the sense sequence, so that a sense segment of messenger RNA 5' adenine adenine guanine guanine uracil cytosine 3' would be blocked by the antisense messenger RNA 3' uracil uracil cytosine cytosine adenine guanine 5' segment. Antisense oligonucleotides have been researched as potential drugs for diseases such as cancers including lung cancer, colorectal carcinoma, pancreatic carcinoma, malignant glioma and melanoma, diabetes, amyotrophic lateral sclerosis ALS, Duchenne muscular dystrophy and diseases such as asthma, arthritis and pouchitis with an inflammatory component. As of 2014, 2 antisense drugs have been approved by the United States Food and Drug Administration, that is US FDA. Formiviricin, marketed as Vitravin, as a treatment for cytomegalovirus retinitis, and Mipomersin, marketed as Kynamro for homozygous familial hypercholesterolemia. Delivery of antisense therapy drugs. Because nucleases that cleave the phosphodiester linkage in DNA are expressed in almost every cell, unmodified DNA molecules are generally degraded before they reach their targets. Therefore, antisense drug candidate molecules are generally modified during the drug discovery phase of their development. Additionally, most targets of antisense therapy are located inside cells, and getting nucleic acids across cell membranes is also difficult. Therefore, most clinical candidates have modified DNA backbones, or the nucleobase or sugar moieties of the nucleotides are altered. Additionally, other molecules may be conjugated to antisense molecules in order to improve their ability to target certain cells or to cross barriers like cell membranes or the blood-brain barrier. There is a very important topic of contemporary importance, that is combinatorial synthesis. Combinatorial chemistry comprises of chemical synthetic methods that make it possible to prepare a large number, tens to thousands or even millions of compounds in a single process. These compound libraries can be made as mixtures, sets of individual compounds, or chemical structures generated by computer software. Combinatorial chemistry can be used for the synthesis of small molecules and for peptides. Strategies that allow identification of useful components of the libraries are also part of combinatorial chemistry. The methods used in combinatorial chemistry are applied outside chemistry too. Synthesis of molecules in a combinatorial fashion can quickly lead to large number of molecules. For example, a molecule with 3 points of diversity R1, R2 and R3 can generate NR1 into NR2 into NR3 possible structures, where NR1, NR2 and NR3 are the numbers of different substituents utilized. The basic principle of combinatorial chemistry is to prepare libraries of very large number of compounds, then identify the useful components of the libraries. Although combinatorial chemistry has only lately been taken up by the industry since the 1990s, its roots can be seen as far back as the 1960s when a researcher at Rockefeller University, Bruce Merrifield, started investigating the solid phase synthesis of peptides. In its modern form, combinatorial chemistry has probably had its biggest impact in the pharmaceutical industry. Researchers attempting to optimize the activity profile of a compound create a library of many different but related compounds. Advances in robotics have led to an industrial approach to combinatorial synthesis, enabling companies to routinely produce over 1 lakh new and unique compounds per year. In order to handle the vast number of structural possibilities, researchers often create a virtual library, a computational enumeration of all possible structures of a given pharmacophore with all available reactants. Such a library can consist of thousands to millions of virtual compounds. The researcher will select a subset of the virtual library for actual synthesis based upon various calculations and criteria such as computational chemistry and QSAR. There are two main approaches in combinatorial chemistry. The first is the split mix method, which involves attaching the starting compounds to polymer beads. The beads are then split into numerous groups, say for example 50, and reacted with the second set of reagents. After the reaction, all the beads are pooled, mixed together and split into numerous groups, say 50 again. If mixing is efficient, each group should contain approximately equal number of beads representing each of the innumerable, here 1000 first generation products. The groups of beads are then reacted with the next set of reagents. If the beads are tagged in some way after each reaction, for example with a different fluorescent label to identify each reagent, the combination of tags will characterize each of the 50,000 second generation products exactly. The finished beads can be screened for their ability to bind a particular target protein in the body. The compound with the best performance can be identified and tested further. Additional rounds of pooling and splitting allow libraries with millions of compounds to be generated and subjected to high throughput screening (HTS). The second method of combinatorial synthesis is called parallel synthesis. Here, all the different chemical structure combinations are prepared separately in parallel using thousands of reaction vessels and a robot programmed to add the appropriate reagents to each one. This method is unsuitable for the creation of very diverse libraries, but is very useful for the development of smaller and more specialized libraries based on a particular skeleton, that is starting compounds. Parallel synthesis is a technique widely used by researchers to speed the discovery of new compounds and to screen for optimal process conditions. In the pharmaceutical industry, parallel synthesis speeds the discovery and development of potential drug candidates. Early practitioners coined the term combinatorial chemistry to describe the synthesis of large libraries with diverse chemical structures screened in mass for potential biological activity. Today, parallel synthesis is available to any researcher who wishes to benefit from the time saving and additional data created by running multiple experiments in parallel. Parallel synthesis applications include lead generation, lead optimization, and screening for optimal reaction conditions. Researchers also optimize processes through better understanding of synthetic routes, solvent systems, optimal temperatures and concentrations, correct reagents, reaction times, and selection of catalysts. Limitations of combinatorial synthesis. Although highly useful, combinatorial synthesis has its own disadvantages. Few of the most common limitations of combinatorial synthesis can be listed as follows. The first one is slower reaction kinetics. Unlike solution phase reactions, a solid support for holding one of the reactants is often employed in case of combinatorial synthesis. This reduces the number of active collisions between the reactants and eventually decreases the reaction kinetics. Second, use of large excess of reactants. For the initial loading as well as the subsequent elongation steps, a large amount of the reactant is needed for overcoming the slower kinetics. Most of the times the excess reactants are not recycled, resulting in a huge loss. Third, use of excess solvents for washing. For a clean library synthesis, a large excess of solvents is used for coupling, deprotection and cleavage. This leads to huge economical loss as well as being unfriendly to the environment. Use of high-cost coupling reagents. For a desired regio and stereo selectivity of the resulting libraries, a huge excess of very costly coupling reagents is needed. This is contrary to traditional solution phase reactions where low-cost coupling reagents in minimum concentrations are needed. Very small amounts of the synthesized compounds are made available through combinatorial synthesis. Preparation of sub-libraries through deconvolution is time-consuming. For insoluble supports, the reactions can't be monitored continuously through spectroscopic techniques. Greener methods of synthesis are of limited applicability for combinatorial reactions. That's all we have in this lecture. Thanks, dear friends.